The overall goal of this technique is to genotype CRISPR Cas9-induced insertion-deletion mutant clones in a high-throughput manner.
This method can help answer key questions in the genetics field, such as the phenotypic effect of knocking out a particular gene in a cell model of choice. The main advantage of this technique is that it is amenable to high-throughput screening and is multiplexable.
Though this method can provide insight into human cancer genetics, it can also be applied to the other systems, such as human, stem, and the primary cell or the cells of non-human origin.
We first had the idea for this method when we performed short tandem repeats or STR typing assays.
To begin the procedure, transfect the chosen cells with a plasmid co-expressing a fluorescent label, Cas9, and the single guide RNA. Trypsinize the cells and select about 3,500 fluorescently labeled clones. Plate the clones in portions of 500, 1,000, and 2,000 cells on 10 cm dishes in 8 milliliters of penicillin streptomycin supplemented growth medium. Incubate the cells in 5% CO2 atmosphere at 37 degrees Celsius, replacing the medium every 5 days until single-cell colonies are visible to the naked eye. Then, aspirate individual colonies into a 200 microliter pipette with about 5 microliters of growth medium. Transfer each colony to a separate well containing 200 microliters of growth medium. Repeatedly pipette each mixture up and down to resuspend the cells.